Artificial Brain Development Project. Functional requirements for a brain reading machine. Measurement of neuron parameters. Resting potential. Threshold potential action. Potential amplitude. Measurement of synapse. Parameters. Synapse type. Excitatory inhibitory. Synaptic strength. Coordinates of presynaptic and postsynaptic parts. Measurement of neurotransmitter parameters. Neurotransmitter. Name neurotransmitter. Concentration. Measurement of receptor parameters. Receptor name. Receptor concentration. Measurement of glial cell. Parameters. Glial cell type. Glial cell function. Assuming we have a frozen brain, how can we use a laser to read the above information, slice the red layer, and remove the remnants? Algorithm of the system operation. Fluorescent labeling. The brain sample is treated with fluorescent markers that specifically bind to target molecules, neurotransmitters, receptors, etc. Laser scanning. A high-resolution laser scanner reads fluorescent signals from the brain surface. The laser beam induces fluorescence from the labels, and detectors capture the signal intensity. The following parameters are analyzed. Resting potential and threshold potential, through fluorescent markers sensitive to ion concentrations. Action potential amplitude, by changes in fluorescent signal sensitive to voltage. Synapse type, through different fluorescent labels for excitatory and inhibitory synapses. Concentration of neurotransmitters and receptors, by the intensity of the respective fluorescent signals. Type and function of glial cells, by specific fluorescent markers. Layer removal. After scanning, the tissue layer is removed using laser ablation. The laser system carefully removes the scanned layer to expose the next tissue layer. The process of scanning and layer removal is repeated for subsequent layers of the brain. Technologies and equipment. Laser scanner. A high-resolution laser for reading fluorescent signals. Detectors to capture fluorescence intensity. Fluorescent markers. Specific markers for neurons, synapses, neurotransmitters, receptors, and glial cells. Laser ablation system. A laser setup for precise slicing and removal of tissue layers. Example workflow. Fluorescent processing. The brain sample is infused with fluorescent markers. Scanning. The laser scans the surface, capturing fluorescent signals and transmitting data to a computer for analysis. Ablation. The laser removes the scanned tissue layer, preparing the next layer for scanning. Analysis and recording. Collected data are analyzed and recorded for further use. Conclusion. This method provides a streamlined and targeted approach to reading parameters from the surface of a frozen brain with subsequent layer removal. The use of fluorescent markers and laser scanning efficiently captures the necessary parameters, while laser ablation ensures precise removal of scanned layers. Copyright 2023. Artificial Brain Development Project. Gdansk. Copyright 2024. Human Artificial Intelligence. Copyright 2024. Anton Hladkovich.